Welcome back, Julia.
Thank you. Thanks for that introduction. So today I'm going to present pretty much the entire work of my master's degree, which I'm just wrapping up now in my third year. And the topic of my master's was Multilocus species delimitation and species tree inference in the western rattlesnake species complex. So first I want to begin with a review of how we can use multilocus data to delimit species. Here we can see a species tree depicted as these gray hollow pipes. And within the hollow pipes of the species tree, we can see independently evolving gene trees. At the tips of the species tree, we see putative species A, B, C, and D. And using this information, we can statistically test whether the independently evolving gene trees show species A and B, for example, as independently evolving, or whether they're more appropriately lumped into species AB. The problem here is that we need to know that A and B are sisters, so we need to know a guide tree. So how do we get a guide tree from multilocus sequence data? Well, if we again start with our independently evolving gene trees, we can lump the terminals of these gene trees into putative species A, B, C, and D again. We can infer the species tree on top of that. The problem here is that we need to know our species groupings before we can do this inference process. So to restate the problem, in order to delimit species using multilocus sequence data, we need to know our species tree, and in order to infer the species tree, we need to know our species limits. So if we don't know either of these, we can't do either of these. The solution that we propose is simultaneous inference of the species tree and the species limits from the multilocus data. So I go about this using a hypothesis testing approach, where our hypotheses are competing models of speciation or competing models of species delimitation here. So a hypothesis testing approach to species delimitation has been taken before by previous researcher teams. Here we can see that all of these previous methods require assumptions to be made, such as assuming that we know a guide tree, or assuming that gene trees have been inferred with certainty. Our hypothesis testing approach begins again by generating competing hypotheses of species limits. We then use Bayesian inference to infer the species tree directly from multilocus sequence data, while also coming up with a goodness of fit for each hypothesis, here quantified as the marginal likelihood. And we can compare these marginal likelihoods using the Bayes factor to allocate statistical support. So what is the marginal likelihood measuring, and what is the Bayes factor telling us? Here we can see that Bayes' theorem provides a framework within which we can estimate parameters theta given existing knowledge of a data set D and a model of evolution M. So this is the posterior distribution. In this equation, in the denominator here, we can see a value representing the probability of the data set D given the model of evolution M. In other words, this is the marginal likelihood of the model M. We can compare this probability of multiple models if the data set D is held constant using the Bayes factor ratio. So here we're comparing the marginal likelihoods of A and B to competing models. And we can log transform this equation to make it useful for allocating statistical support. So as with using Bayes' theorem for practical purposes in phylogenetic analysis, we have to use Markov chain Monte Carlo to estimate that. We also need to estimate the marginal likelihood. So here we can see three competing estimators of marginal likelihood and comparisons of how they work and the advantages and disadvantages of each. The most widely implemented estimator of marginal likelihood has historically been the harmonic mean estimator. Here the harmonic mean estimator samples from the posterior distribution to calculate the harmonic mean of likelihoods in the posterior distribution. And the smooth harmonic mean estimator just also samples from the prior distribution to stabilize that value. This has been widely implemented in the past because it's fast computationally and very easy to implement, but has been shown to provide inflated likelihood scores and can have infinite variance even under very simple circumstances. So this is a very inaccurate estimator of marginal likelihood here. Thermodynamic integration implemented via path sampling and the stepping stone estimator both have become more computationally feasible recently. These require drawing a second distribution relating the posterior distribution back to the prior, and path sampling integrates over this resulting distribution, while the stepping stone method subsamples from this resulting distribution. Both of these have been shown to be highly accurate estimators of marginal likelihood, and stepping stone is slightly less computationally intensive. So now we are able to more accurately estimate marginal likelihood and apply these Bayes factor methods. So the objectives of my study are to demonstrate this method of simultaneous species delimitation and species tree inference, and I want to demonstrate this in a model system. A model system here will have both the species limits unknown and the species tree unknown. So the western rattlesnake species complex is a perfect candidate model system to apply this methodology. Here we can see the 9 subspecies within the western rattlesnake species complex. This slide illustrates the extreme morphological diversity found within this complex. Just 15 years ago, all of these subspecies were grouped into one widespread polytypic species, and since then, taxonomic revision has been rampant within the group. So species limits are very contentious. Further, this study is the first multilocus study of this species complex, so the species tree is unknown. Here we can see the wide range of the 9 subspecies within the Crotalus viridis, or western rattlesnake species complex. All 9 of these subspecies are currently grouped into 3 species. First, we have Crotalus viridis, shown in red and orange. Next, Crotalus cerberus, shown in gray. And all remaining colors on the map fall into Crotalus origanus. The only previous molecular work done in this group was carried out over a decade ago. 3 nearly concurrent mitochondrial studies all came up with the same general reciprocally monophyletic groupings in their mitochondrial gene trees. I've highlighted those reciprocally monophyletic clades here, and they all differed in their taxonomic recommendations, though they found the same result. So current taxonomy recognizes these 3 reciprocally monophyletic mitochondrial clades as the 3 species within the group. Again, Crotalus viridis in red, sister to all remaining. Crotalus cerberus in gray, sister to Crotalus origanus in green. Okay, on to my methods. First, I want to outline my methods before I dive right in. First, I'll talk about data collection and gene tree inference. Next, I'll walk through my methodology for simultaneous species delimitation and species tree inference. Next, I apply BPP for comparison. BPP is a widely implemented multilocus Bayesian species delimitation methodology. And I use my resulting species delimitations to construct the first dated multilocus phylogeny of the Crotalus viridis complex. Shown here are the 63 individuals sampled for my study. All of these individuals were loaned to me by various museums and individuals, including the MBZ. I've also included 2 outgroup species, Crotalus scutellatus and Crotalus adamantius. 8 of 9 subspecies were included in this study. The only missing subspecies is here, Crotalus origanus caligenus. This island endemic has been shown to be mitochondrially nested within the blue Crotalus origanus helleri. So lack of sampling here is detrimental, but not the worst thing in the world. Okay, so on this map too, I want to point out some notable sampling gaps, because there are some. First, there's large sampling gaps within Crotalus viridis in red. There's a large sampling gap throughout the entire center of the Crotalus origanus lutosus range. And there's a large sampling gap throughout northern California of Crotalus origanus. And the flaws of this study are notable in the sampling gap. Finally, I want to point out that these 3 individuals highlighted in orange are my 3 individuals of Crotalus viridis nuntius, the range of which is shown in orange, and they all fall outside of the range of this subspecies, but were morphologically and molecularly verified to be of this subspecies. I sequenced 5 nuclear introns and 1 mitochondrial coding gene for these individuals using standard Sanger sequencing methods. Sequences were aligned in muscle. Haplotypes of nuclear introns were inferred using phase. And I checked for recombination using topology. For gene tree inference, I inferred the gene trees using both a maximum likelihood and Bayesian approach using the parameters shown here, and all of these gene tree inference analyses were repeated twice. Now I want to walk through the methodology of our simultaneous species delimitation and species tree inference. So the first step here is that we have to generate competing hypotheses of species delimitation. The second step, this is a review slide from the introduction, by the way. The second step is Bayesian inference, and the third step is comparing the fits of these hypotheses. So for step 1, hypothesis generation, hypotheses were generated in 3 ways. First, historic subspecies were treated as species. Second, I collapsed nodes on the mitochondrial gene tree. And third, I used multilocus nuclear clustering implemented via POFAD to look for any additional genetic clusters. So for method 1, I applied the subspecific designations that were used by the loaning institution, and if anything was equivocal or unavailable, additional information such as mitochondrial data or morphology or locality information was used to supplement this information. For method 2, I started with the ND2 Raxamal-inferred tree. I grouped clades on this tree into strongly supported, reciprocally monophyletic mitochondrial clades. And on this resulting tree, I iteratively collapsed nodes within the tree to create increasingly inclusive, nested and non-nested hypotheses of species delimitation. For method 3, I used POFAD here. POFAD takes as input multiple single locus distance matrices that are relating alleles and creates one multi-locus distance matrix relating individuals. For my single locus distance matrices, I used POUT, and the results of the POFAD analysis were visualized using splits tree. The second step of the simultaneous inference methodology is Bayesian species tree inference. All Bayesian species tree inference was carried out in StarBeast, implemented using Beast with Beagle. All StarBeast analyses differed only by the a priori species designations, so here that is the input traits file for StarBeast analyses. All of these analyses were repeated with and without mitochondrial data included. And the parameters shown here were applied for all StarBeast analyses. And again, all StarBeast analyses were repeated twice. To simultaneously estimate goodness of fit for each of these StarBeast analyses, I estimated marginal likelihood using 3 competing estimators, the 3 that we talked about in the introduction: the smooth harmonic mean estimator, path sampling, and stepping stone. For estimation using the smooth harmonic mean estimator, the harmonic mean was calculated from samples of likelihoods drawn from both the posterior and prior distributions. For both path sampling and stepping stone, I inferred a secondary distribution of 100 power posteriors that related the posterior distribution back to the prior. For path sampling, the likelihood was integrated over this resulting distribution, and for stepping stone, samples were drawn from this distribution, and the likelihood was estimated from this subsample. And all code for this marginal likelihood estimation is credited to Beal et al. 2012 and was run in Beast. So now that we have our goodness of fit generated, I compared these goodness of fits using the Bayes factor. So first, I identified the best fitting hypothesis, meaning whichever hypothesis had the highest estimated marginal likelihood under each estimator. And because the Bayes factor is a pairwise comparison, I pairwise compared all other hypotheses to this best fitting hypothesis. And log transformed this to apply statistical support. So in accordance with Cass and Raftery, 1995, 2 ln base factor support of greater than 10 is considered very strong. So this was my cutoff for species delimitation. So for comparison, I also applied BPP. BPP uses a reverse jump Markov chain Monte Carlo algorithm. That algorithm allows the number of parameters theta to change during Markov chain moves. So this is a type of move that's not allowed under classical Markov chain Monte Carlo. So this allows for nodes to be collapsed and resolved during Markov chain moves. And that means that we need to know our nodes, which means this requires a guide tree. So to illustrate this, we see sisters C and D here. BPP will test whether C and D should be collapsed into CD, and then we'll test whether B, C, and D should be collapsed into BCD. But it can't test anything that's not node-based. Because the species tree is unknown within the Crotalus viridis species complex, I reran BPP using 2 separate starting trees. First, I applied the mitochondrial gene tree as my starting tree, and next I applied the Multilocus species tree if it was found to differ in topology from the mitochondrial gene tree. All BPP analyses were repeated with and without mitochondrial data included. BPP was repeated using both algorithms and multiple priors under each algorithm, and the priors shown here were applied as fixed priors to all BPP analyses. Finally, I inferred a dated phylogeny of the Crotalus viridis complex. For a priori species designations for this dated phylogeny, I applied the best fitting hypothesis of species delimitation recovered using the base factor method. And I applied a 0.65% changes per million years rate of evolution to ND2, and all other rates were estimated based on this ND2 rate. So any discussion of species limits and species tree inference requires a discussion of species concepts. The assumption of all coalescent-based methods of both species delimitation and species tree inference is that all gene tree discordance is due entirely to incomplete lineage sorting. So that means that the methods shown here, as well as BPP, operate on the assumption that if there's enough gene flow between species A and B, the method should favor lumping them into species AB. So this is directly compatible with the evolutionary species concept, which defines a species as an independently evolving metapopulation lineage. Okay, so for my results, I first want to briefly discuss a summary of my data collection shown here. So the data set that I used was 64% complete, and nuclear introns ranged from having 4 to 35 in-group parsimony informative sites. I briefly want to talk about some trends I saw in the nuclear gene trees. Though there was no reciprocal monophyly recovered for any of my subspecies, I did find some subspecific groupings in a few of my gene trees. In BZW1, I found a cluster of Crotalus organus lutosus from Idaho and Utah. In my RPS8 gene tree, I found a cluster of Crotalus organus just from Oregon and Washington, and I found a cluster of Crotalus cerberus as well. And in my TBP2 gene tree, I found a cluster of Crotalus viridis plus Nuntius, and also another cluster of Crotalus organus lutosus here. So now I want to walk through my mitochondrial gene tree. The results, the mitochondrial gene tree was generally concordant with previous mitochondrial studies. I found a clade of Crotalus viridis, sister to all remaining in-group subspecies. Within that clade was a monophyletic Crotalus viridis nuntius. I found a clade of Crotalus cerberus that had that was weakly supported as monophyletic, but had some deeper structure that did not show any geographic associations. Within my Crotalus organus clade, I again found weak support for the monophyly of this group, and I found deeper structure that did have a geographic basis. So the two clades I recovered within Crotalus organus organus represented a just Oregon and Washington clade, and then a California clade. The California clade has more samples in it here. Next, I found Crotalus organus helleri, sister to, that's hard to see, sorry, Crotalus organus lutosus, and this lutosus clade contained both Crotalus organus abyssus and concolor, though I only had one individual of each of these subspecies represented in this study. So largely concordant with previous mitochondrial studies, except because of my sampling, I was able to detect deeper structure in Cerberus and Organus, gray and green. Okay. Okay, so next I'm going to walk through the results of my hypothesis generation. For generating my hypothesis of subspecies as species, all subspecific designation was unequivocal except for one individual, which was originally designated as Crotalus organus concolor. With the assistance of the California Academy of Sciences curatorial staff, we redesignated this individual morphologically as Crotalus viridis nuntius. Okay, so for my second method of hypothesis generation, I start with my RAX-ML ND2 mitochondrial gene tree. So this is the same tree that we were looking at a couple of slides ago. RAX-ML mitochondrial gene tree. Now the boxed clades here represent reciprocally monophyletic clades that were supported with strong support in both the Bayesian and maximum likelihood gene trees. So notice that within Cerberus and Organus, those have two strongly supported reciprocally monophyletic clades in them each. The lettered nodes here were iteratively collapsed to create both nested and non-nested hypotheses of species delimitation, to create a total of 12 hypotheses based on this method. So to illustrate that, if we collapse node F, we are left with 1, 2, 3, 4, 5, 6 total species, grouping helleri and lutosus into one putative species. If we collapse D per se, a similar arrangement with 6 species, but this is not a nested hypothesis, it's an unrelated hypothesis. We can also collapse both of them at the same time, or we can even collapse a deeper node for a 2-species hypothesis, and so on to create 12 hypotheses based on this tree. So for method 3 of hypothesis generation, I didn't find any clusters, very obviously. So this is a big mess, and no hypotheses were generated based on POFAD. Okay, so here are the 14 total hypotheses generated. Hypotheses 1 through 12 represent those generated from the RAX-ML starting tree. Hypothesis 13 is an additional taxonomic recommendation from a previous study. And hypothesis 14 represents subspecies as species, or method 1. So now that we have these hypotheses generated, the next step is Bayesian species tree inference. So here we can see the species tree inferred under the most split hypothesis of method 2. So essentially, the boxed clades on the RAX-ML tree were used as putative species here. I want to walk through the topology of this tree before I talk about statistical support. So first, we find strong support for the monophyly of the Crotalus viridis species complex. Oh, before I walk further, I want to point out that the values of the nodes are ranges because these are 14 different species trees, and these are the ranges of posterior probability that occurred on all of those 14. So hypothesis 3 would have 0.97, hypothesis 8 had 0.99, etc. for that node there. So again, the monophyly of the viridis complex was supported with strong support in all analyses. Next, we have a surprising result. Crotalus viridis was found to be sister to Crotalus cerberus, so this is contrary to the mitochondrial gene tree, though this node is weakly supported. In all analyses where Cerberus was split into its 2 sister clades, they fell out as sister to one another with posterior probability of 1. I found a clade containing Crotalus organus, lutosus, and helleri. But within this clade, our two clades of Crotalus origanus are not even each other's closest relatives. This is an incredibly surprising find here. So this is the northern Pacific rattlesnake, and the two clades here represent Oregon and Washington and California. Very surprising result. Finally, I find that Origanus B, the Origanus from California, is sister to Helleri, which is the southern Pacific rattlesnake, also in California, Southern California. Okay. So now that we've looked at the topology here, let's look at the marginal likelihood that was estimated for each of these hypotheses. So this is the marginal likelihood estimation using all the data. Immediately we see that the smooth harmonic mean estimator has provided incredibly inflated results. So this is exactly along with expectations based on previous studies. We find that path sampling and stepping stone have found very similar results to one another and support hypothesis 9, which we'll talk about in more detail in a minute, and that the smooth harmonic mean estimator, our inaccurate estimator, is favoring hypothesis 1, treating the whole complex as one widespread species. If we exclude mitochondrial data, we still find that the smooth harmonic mean estimator is providing a greatly inflated result. And here, path sampling and stepping stone are favoring hypothesis 12. So now that we've found which hypothesis is favored with all the data and then with just nuclear data, let's see how much better they are than the rest of the hypotheses using the Bayes factor. So here we can see the Bayes factor results comparing in the all data column, comparing hypothesis 9 to all other hypotheses, and in the nuclear only column, comparing hypothesis 12 to all other hypotheses. So the takeaway point from looking at the numbers in this table is that they're all well above 10. So hypothesis 9 is very strongly favored when all the data are used. Hypothesis 12 is very strongly favored when nuclear data are used only. So let's compare hypothesis 9 and hypothesis 12. Here we can see the species trees inferred under each of those hypotheses. The only difference between these hypotheses is right here. So when mitochondrial data are excluded, there's a tendency to split Cerberus into two putative species. Because I didn't find any strong topological discordance between species trees inferred using all the data and those inferred with just nuclear data, I favor the all data hypothesis 9 here to recommend 6 species within the Crotalus viridis complex: Crotalus viridis, Cerberus, Origanus A, the northern Origanus, Lutosus, Origanus B, and Helleri. So let's see what BPP has to say. All BPP analyses yielded the exact same results, so that's regardless of the starting tree, regardless of which algorithm, regardless of whether mitochondrial data were excluded, and that result is that all nodes were strongly supported except for the one leading to Crotalus Cerberus A and Crotalus Cerberus B. So BPP also favors hypothesis 9. So using hypothesis 9 as my a priori species designations for inference of a dated phylogeny, we see the species phylogeny shown here, and all divergences within the Crotalus viridis species complex fall within the Pliocene or Pleistocene here. For my discussion. Before I discuss my results, I want to briefly discuss the role of coalescent species delimitation in taxonomy and in species tree inference. So if a cryptic species is detected and other lines of evidence are not yet available that unify individuals of this species into a species, are they any less of a species or of an independently evolving lineage? Bauer et al. 2011 argue that descriptive unifying characters are needed for species to be described. But Fujita and Lachey 2011 argue that Bayesian species delimitation removes the subjective lens associated with noting morphological differences. So my study perfectly illustrates how coalescent species delimitation can make the process of species delimitation more objective. In this study, morphological distinctiveness has been previously associated with subspecies within the Crotalus viridis complex. I found support that many of these subspecies are independently evolving metapopulation lineages or species here, and I found just as much support that identifying cryptic species within this species complex, here splitting historic subspecies Crotalus origanus origanus into two putative species. So it would be biased to elevate the previously recognized subspecies without elevating cryptic species as well, just because they have morphologically unifying traits already recorded. For the Bayes factor has previously been used for model selection in a number of scenarios. Here, our model is consists of prior parameter restrictions on Bayesian phylogenetic analysis. So this model includes things like substitution and clock models, demographic parameters, and also includes a priori species groupings. So if only the a priori species groupings vary between these analyses, then any change of fit is due only to the change in these a priori species designations. So if this fit is known with certainty, if the marginal likelihood is known with certainty, this is a very dependable method of species delimitation. And the problem here is that we have to estimate the marginal likelihood. Previously, our smooth harmonic mean estimator, which was one of the only methods ever used for this, made this very problematic. My study recovered similar highly inflated likelihood scores estimated under the smooth harmonic mean estimator, and found results that differed dramatically from path sampling and stepping stone estimates. I also found that path sampling and stepping stone provided similar scores as one another, which was also expected. And now that we have this more accurate estimator of path sampling and stepping stone that are computationally possible, we can accurately estimate marginal likelihood and accurately use the Bayes factor. To compare this with BPP, let's look at sister species B and D on the true tree on the bottom. So in the guide tree, the node leading to B and D does not exist. So collapsing this node cannot be tested. And even if B and D are exchanging genes readily, B and D would be supported as separate species if the inaccurate guide tree was applied for BPP analyses. So this is how BPP can lead to oversplitting if an inaccurate guide tree is implemented. The Bayes factor method demonstrated here does not rely on a fixed guide tree. So this topological constraint is removed. So if B and D are within the analysis at all, they can be placed as sister, and other additional hypotheses can be tested based on these results. Now I want to discuss the taxonomic implications of my study here. I recovered support for 6 species within the Crotalus viridis complex, and I found the mitochondrial gene tree was not topologically concordant with the multilocus phylogeny. So this underscores once again that multiple independently evolving loci must be included for accurate phylogenetic estimation. Here, the mitochondrial gene tree differed from the multilocus phylogeny in that Crotalus viridis was found to be sister to Crotalus cerebrus, though with weak support again. Crotalus origanus A and Crotalus origanus B were not found to be each other's closest relatives. And Crotalus origanus B was found to be sister to Crotalus helleri. So let's look at these 6 species on the range map once again to get an idea of where they're falling. We first see Crotalus cerebrus, sister to Crotalus viridis here. Next, our Crotalus origanus A represents samples just from Oregon and Washington. Crotalus lutosus, even though I had a large sampling gap in the center of this range, all of these individuals fell into a strongly supported mitochondrial clade. Crotalus helleri here is found to be sister to Crotalus origanus B. And given these new results, I would say that my most notable sampling gap here is the lack of samples from this region here. That would help us delineate these a little bit better. Okay. So based on these data, I have some taxonomic recommendations, and I want to say that taxonomic recommendation here is, the purpose is to encourage that taxonomy better reflect true species diversity and evolutionary history within the viridis complex. So I recommend that Crotalus cerberus remain as it's currently designated, the Arizona black rattlesnake. I recommend that Crotalus helleri contain the southern Pacific rattlesnake, as well as the Coronado Island rattlesnake. This individual was not represented in my study, but previous mitochondrial studies again have found this island endemic to be nested within Crotalus helleri in previous mitochondrial studies. I recommend that Crotalus lutosus contain the Great Basin rattlesnake, midget faded rattlesnake, and the Grand Canyon rattlesnake, though these were only represented by a single individual in my study, these latter two. So additional systematic work is paramount to making any recommendations with confidence here. I recommend that the species name Crotalus origanus be applied to the northern Pacific rattlesnake from Oregon and Washington, as the type locality of Crotalus origanus is from Oregon. I recommend that the Crotalus origanus from California be renamed based on this. And I continue to recognize the prairie rattlesnake and the Hopi rattlesnake within Crotalus viridis. So to conclude, I have successfully demonstrated this method of simultaneous species tree inference and species delimitation that uses the Bayes factor. This approach incorporates uncertainty in gene tree estimation, since we're starting with multi-locus data, not with gene trees. Here we don't need an input guide tree. And we have statistical support to compare non-nested models of species delimitation. I have reassessed the understanding of the phylogeny within the Crotalus viridis species complex, and as a result, recommend taxonomic revision that better reflects the evolutionary history within this group. So I want to leave with the thought that species limits and speciation history are interlaced, and it's inappropriate to study them independently. They must always be studied together. And as I'm at my, so thank you all for listening to my talk. I want to thank all my learning institutions here, especially the MVZ, because I got all of my California origanus samples from the MVZ. So you guys helped discover a cryptic species potentially. I thank my funding sources, my fantastic advisor, and happy to be back at Cal, so go Bears.
That was a great talk, really cool research. Just wondering, do you plan on describing those species, doing those descriptions, and whether you are or not, how would you recommend actually doing the species description given the kind of data you used?
Right, so for all of the species descriptions where morphological work has already been done to allocate these into subspecies, that's not a problem. For, I think the main question is probably about the northern Pacific rattlesnake. Finer scale sampling is incredibly necessary to find out where that line actually is between the two. I wish that I hadn't discovered that at the very end of my master's. But I think...
There's your PhD.
Yeah, I would love that.
Would you be okay with a species description that was just based on molecular characters, for example?
I would. Yeah, I think that is completely fine, in my opinion. I'm on that side of the debate, for sure. Yes.
So thank you for a really cool and really nicely presented talk. I'm wondering about the California gap, since a lot of us do, pick up just go herping for fun. Where's your northern gap and where's your southern gap in California? Like, is the Bay Area part of the gap, or?
So I received samples from just north of the San Francisco Bay, and that's it. And then all the rest of California
So-
is blank.
So can I ask why you didn't get samples from Cal Academy? Because I know I've collected rattlesnakes from Mendocino County and a couple of other places up in Northern California, and those were all tissued, and those should be at the Cal Academy.
Well,
Is there a reason?
I requested whatever they had available for tissues, and I wasn't aware of those samples.
Okay. We should talk to Jens again.
Yeah. Well, that's unfortunate. Okay, so so this is the large gap here, I guess, and here's the bay.
Yeah, the Stony Creek site has a couple of rattlesnakes.
Okay, I'll look into it. Yes.
So you have nuclear genes, mitochondrial genes, and morphology in your talk, but you don't have any ecology. And there's a lot of interest now in ecological species formation. For example, I would make a case that that Coalescent might be an ecological species because there are other very distinctive forms out on Coronado Islands. So what if Coalescent is an ecological species, or what if let's say Abyssus is another possibility as an ecological species? If you use strictly a molecular approach, aren't you going to miss all of that?
I think that boils back to again species concepts, and this study is essentially about how we can use genes to detect species differentiation. So I feel like that is out of the scope of where my experiences lie.
But you're proposing to make a taxonomic judgment, and I say then that taxonomy is out of your realm.
I would agree. Yeah.
I have two. One is a point, in case you do further studies, we do have tissue frozen upstairs of a Coronado Island rattlesnake. It's not online yet, but Dave and I were on the trip and we collected it. I've seen it in the freezer within the last month, so if you do need it to verify or contradict what Dave is suggesting, it's just a waiting.
I'm very interested in that.
But my argument would be that she couldn't contradict it.
Yeah. And the question is, you know, pointing out this gap that you have basically from the Bay Area to Oregon. There are dozens, if not hundreds, of preserved specimens in museum collections. Have you thought of looking at, perhaps nobody's looked carefully for morphological
Right.
differences. In fact, then you could combine your molecular work with perhaps distinct
It
morphological.
has been shown time and again that once cryptic species seem to be discovered molecularly, then you look closer and find morphological uniformity.
Yeah, I'm doing the same thing right now with the California legless lizards, where they're molecularly quite different, and I'm desperately trying to find ways to tell them apart.
Are you doing toe counts, Ted?
I'm waiting for them to re-evolve limbs. It will be a while, so don't hold your breath.
I believe you've had your hand up.
Well, I'm trying to ask a similar question, but I wanted to ask another one as well. Are there any names that are already available that are buried in the synonymy of Oregonus
I haven't been
that
able-
would
in
apply
California?
to the southern or the northern, whatever you?
In my, I am currently looking more extensively with the assistance of the San Diego Natural History Museum, but I haven't been able to find much.
Well, I mean, there must be in cloudbird rattlesnakes, there must be a
Right.
list of all the synonyms that are, whoever did
Right.
the last revision.
I haven't been able
So
to
there
find
might be a
anything
name
that
already
has,
available for that taxon.
there may be. I haven't been able to find it yet because California snakes that were found then were called Organus after the northern ones and things like that. So I'm still looking. Yes.
So I was interested to see your results regarding abyssus and concolor because they often come out closer to viridis, and essentially they overlap with viridis in a lot of their areas. Do you feel confident in your assessment as putting them as a subspecies of Lutosis, or what would you do if you went on to describe these species?
Previous mitochondrial studies have placed both of those that have had more extensive sampling have placed both of those near lutosis in the tree, somewhere around lutosis or helleri, far from viridis. So even though the ranges are close, this is definitely a secondary contact. And even though, for example, both of these are dwarf taxa, that looks like convergence. So I'm confident that those individuals are near lutosis, but I'm wary that they're within.
Nice research. So my question is related to like many other questions that people have asked, and basically it's like when you find you said that it's okay to describe the species with molecular data, and that's fine. But my question is, how would you actually diagnose those? Because you have like some kind of discordance between, at least some, between your mitochondrial data and your molecular data, nuclear data, and then most of the genes, the nuclear genes that you showed, don't have obviously your clades, right? So how you actually diagnose those based on the current data that you have?
Right. So the majority of these are diagnosable morphologically and geographically based on subspecies. The only pair that don't currently have that diagnosis are these two clades within Origanus. So again, looking closer for those two. But mitochondrially, the two clades of Origanus were very divergent, deeply divergent, and strongly supported as reciprocally monophyletic. So I would be confident in that molecular diagnosis. I don't have the morphology to go with that yet.
I was impressed with your analysis, but I had a question about the Bayesian support for lutosis, the two Origanus and helleri, because none of those were
In the species tree?
yes, none of those were. I think the highest was 87, posterior probability of 87.
Right. The monophyly of the
seems-
group was supported, but within it was equivocal.
Right. The monophyly was definitely supported. Yes. Right. So there you get it. So the monophyly is supported at 94 to 100, but the highest, well, you've got a 91 up there, but there's not much. Those are kind of low posterior probabilities.
Right. I agree that the interrelationships within that clade are definitely equivocal, and would hope that the addition of more data would resolve that.
Now, I think it's really critical to get some more data on that.
Well, I have a question about that, but that's a different issue, right? I mean, so that's, what are the interrelationships of these inferred lineages, but the analysis that you were doing to identify species is actually for the individual clades themselves, for
That's
helleri,
true.
for Origanus B, et cetera.
Right.
For lineage independence, that's right.
Well, what do you think? What do you think additional sampling would do? I mean, what if? I mean, I can't. It's hard for me to understand exactly what might happen if you filled that gap between Origanus A and B. What possibilities might there be?
I don't expect that they're exchanging genes there because they were so deeply divergent in the mitochondrial gene tree. So I don't expect that increased sampling would convolute anything. I
But
feel
as you
that
get closer and closer, they might be exchanging genes. I mean, how
it's obviously a possibility.
over what? In other words, over what geographic scale would gene exchange have to be limited before you would accept that they are the same species?
I haven't considered that because I haven't found any evidence of gene flow between them yet.
But you've got several 100-mile gap.
Right, as I do within the entire lutosis clade, and I found lots of gene exchange within lutosis. So I'm fairly confident that increased sampling within that gap wouldn't lead to any big surprises.
The critical reason for closing that gap is so you can define the boundaries of those two
Right.
things that are apparently morphologically identical. And so
Right.
anybody who's out in the field or otherwise comes into contact with one of those two species has got to know where that boundary is.
Right.
The other, I mean, I'm a mammologist, so but I only think about these snakes in terms of what their what they prey on. They prey on dominantly are California ground squirrels. And the interesting thing there is that California ground squirrels also have this Bay Area to the south and northern deep clade structure. Those things are now being pulled apart as different species. And those snakes and the ground squirrels interact in interesting ways because some populations of ground squirrels are immune to the snake venom and behave to the snakes quite differently than others. And that is apparently a northern southern species boundary issue for the ground squirrels. So I wonder if there's not differences in the venoms of this Oregonus A and Oregonus B clades that you might be able to use also to help diagnose these things.
Phenomics would definitely help. I don't think anybody's looked at any sort of difference between north and south there in that respect. So that's really exciting to hear. Yeah. Yes.
Piece of sort of working around. The question I have is, you know, your species concept is sort of absolute. It's a box, right? And the more tools we get, the more we find that things don't split that exactly in the box. And so how do you make the decisions where you have a bit of interbreeding, but that morphologically, behaviorally, ecologically, they maintain differences, you know, I mean, it always bothers me that we've got two systems here, one which is absolute and another one which is not, and so which wins? I mean, it seems to me the real world results have to win, and we have to find a way of dealing with the absolute boundaries, the species concept.
That's why I like the evolutionary species concept so much, because it maintains that if a group is evolving on its own trajectory, then that is sufficient. And this method tests for whether there's gene flow that's convoluting their ability to evolve independently of one another. And that independent evolution can be due to ecology, it can be due to some kind of biological barrier, or any number of things. So,
As objective as we can try to make species delimitation, the reality is there's always going to be some level of subjectivity. And I think taxonomists get unfairly criticized for being too subjective, when in fact, you know, what is the 5% p-value in ecology? It's a subjective threshold. So, I mean, there is no absolute, you know.
Well, I think as we go along, we see that, yeah, there in nature there are lots of absolute good species that hold up no matter what
Sure.
test you apply to them, but then there are these problem ones where, I mean, I work with deer. Okay, so you take something like a Sika deer in Asia and a red deer in Europe, and you dump them together. Obviously, they've been isolated for who many knows how long, and then you have interbreeding. Well, does that make them the same species? Well, that throws out so much, you know, that I find that getting rid of it is a bigger problem than dealing with the fact that the boundary between the two is not absolute, and if humans hadn't filled around with them, we would have never had a problem.
Okay, on that note, so clearly a talk on species delimitation is likely to engender an hours-long debate, and we've started along that path. I think we've held Julie's feet to the fire for far too long already, so let's thank her again for a really nice conversation.
That's a really nice study.
thank you.
It's very provocative.
Yeah, it's been a little controversial.